DNA extraction and purification involves three basic steps: breaking the cell to expose the DNA, removing the membrane lipids through the use of an appropriate detergent, and precipitating the DNA with alcohol. There are also two optional steps that are almost always done prior to the precipitation step: the removal of proteins by adding the appropriate protease, and the removal of RNA with the use of an RNase. RNA extraction and purification, on the other hand, involves four basic steps. First, you need to disrupt the cells by adding quinidine thiosinate and a reducing agent to the sample, and then subject it to vigorous shaking or vortexing. This step will also break the disulfide bonds and inactivate the contaminant proteins present in the sample. After cell lysis, you need to add phenyl and chloroform isoamyl alcohol to separate your RNA sample from the solution. The aqueous phase contains your previous RNA, so you need to put it in a separate tube. Add isopropanol and centrifugate the solution to precipitate your RNA. Finally, washing the precipitate with 75% ethanol will remove any impurities from the sample. Now, the following chart shows different methods for extracting genomic DNA from Lactobacillus, Arabidopsis thaliana, and saliva. In the chart, we can clearly see that the isolation of DNA from plant and animal cells is different from bacterial cells because of the conformation of their outer membranes. Animal cells have membranes composed of phospholipids, so its degradation involves the use of detergents or chemicals that are able to break it. On the other hand, bacterial cell walls are made of peptidoglycans, so enzymes are needed to break them. For example, bacterial cell wall degrading enzymes lysozyme. Plant genomic DNA is more difficult to extract because of the plant cell's wall, which is removed by homogenization or by adding cellulose to degrade the cellulose that make up the cell wall. Nowadays, most labs use commercial DNA extraction kits. DNA extraction kits which employ spin columns for isolation of the DNA or RNA. The spin columns contain a silica resin that selectively binds DNA or RNA depending on the cell conditions or other factors influenced by the extraction method. These kits follow four basic steps. The first step in RNA and DNA extraction is lysis. The lysis formulas may vary based on whatever you want to extract, DNA or RNA. The second step is purification, or binding the DNA or RNA to the column. In this step, ethanol is added because it influences the binding. The third step is washing the DNA or RNA for removing impurities. The fourth and last step is RNA and DNA extraction, or elution. The final step in DNA extraction protocol is the release of pure DNA or RNA from the silica. After isolation and purification of nucleic acids, it is necessary to quantify their concentrations in the sample. Among the quantification methods, we can find the following ones: quantification with a spectrophotometer, quantification by ethylene bromide fluorescence emission. Here, fluorophores bind to DNA or RNA. This method compares fluorescence intensities to estimate the nucleic acid concentration. Another method is by the use of HPLC columns. This method uses a specific column to separate RNA and DNA according to their characteristics, such as weight, affinity, size, polarity, etc. Another method is the use of Southern blot method that uses specific probes which will be directed target to sequences in nucleic acid sample.